Hello, everyone. I am Aleksandr G. Refeld. My lecture for today is Intestinal Immunity, the brief overview of the field. Okay, just for start, I suited up properly for the lecture. The guy is on the closet, as you can see, toilet. Okay, intestinal immunity. The film speaks up. Why is it interesting? Because the intestine has some unique populations of cells and some unique properties that are not very common through other tissues. Because intestine is the extra organism environment, but inside our cells. So we have the lung epithelial, which is the same, but the insemination of microorganisms in this epithelia is far less than in the intestine. And we have skin, but okay, skin is outside. It's not inside of our body. And let's start from the beginning. It doesn't work. Okay. It's off. Okay, now it should be. Yeah. The components of the intestinal immunity are usual for the first time. The gut epithelial cells, which are at the border of the intestinal, intestinal, oh my god, no, intestinal, not wrong button, intestinal epithelia and the intestinal lumen. The stroma, which comprises the stromal cells, which builds the construction of the epithelia, and it contains some innate immune cells, then the lymphocytes, some more lymphocytes, some more innate immune cells, and some nerve regulation. And we can divide the innate immunity and the adaptive immunity into two subgroups. It's the cellular and humoral. When we're talking about the innate immunity, the cellular immunity, it's the innate immune cells, and the humoral immunity, it's some kind of antibacterial, antimicrobial peptides. And the humoral immunity, in the case of adaptive immunity, that's antibodies. Let's start from intestinal epithelial cells. What do they do? They're not the immune cells in the common meaning, of course, but they produce the mucosa, mucin, that's the slime which makes the digested food move forward. It's like the lubricant, and of course, it prevents the microbial adhesion at the epithelium and prevents the progression of some pathogenic microorganisms. And the intestinal epithelia has two large groups of cells. It's the common intestinal epithelial cells and the M cells. Common intestinal epithelial cells help us assimilate some digested parts of our food, the proteins, glucose, and some antigens that are harmless. And we should be tolerated to it. We shouldn't react to it, because the excessive inflammation is always bad. But the M cells, they make some process that is called transcytosis, and they transfer the potentially harmful antigens from the lumen of the intestine to the Peyer's patch. Peyer's patches, it's the dispersed epithelial-like tissue that provides the antigen presentation. So when we are talking about the antigens that are potentially harmful, they are transferred from the lumen to the Peyer's patches by the M cells. And when we are talking about the harmless antigens, they are transferred through the common epithelial cells. And that's very important, because if you're starting to transfer harmless antigens through the M cells, that's the cause of the food allergy. because your body will react with the immune response, with excessive inflammation on something that cannot harm you. Then the innate and adaptive immunity in general. In general, they are the same as all the tissues in our body. It comprises of innate immune cells, which are basophil cells, eosinophils, neutrophils. Neutrophils are the first line of defense. They move the first to the site of any inflammation. Eosinophils are predominantly fight the parasites. Basophil and mast cells. Well, functionally, they are very similar. They have different origins, but functionally they're kind of similar. They induce inflammation. And neutrophil cells, which are originated from the common lymphoid progenitor, so they are from the lymphoid lineage, but their functions is less like in the field of innate immunity. Macrophage and dendritic cells, which are phagocytic cells, and they can present the antigens to the lymphoid cells of adaptive immunity, which are T cells and B cells. And the B cells and T cells can provide the specific immunity. So, in general, innate immunity, I'm sorry if you know this, but I have to do it because immunity is not very easy to understand. These things recognize some conservative antigens, some antigens that were co-evolution and that evolution with the host, with our immune system for the millions of years. Especially that's usually some parts of microbial cells that they cannot just change with a snap of fingers. The components of the cell wall, they're proteins which are different in sequence than ours. They're some kind of lipids and phosphorylated proteins, etc., etc. And this immune response is not very finely related. It's just the drudge inflammation. And this response is like the surgeon. It's very specific. It can be, it can react on the one certain antigen, because the B cells and T cells suffer some somatic recombination and hypermutation in the case of B cells. And of course, some antimicrobial peptides. I won't concentrate on them. You just have to know that they are exist. And well, they're not just antimicrobial, they have some regulatory properties, but okay, that's not very sufficient. Some important stuff. The T cells generally have two main subgroups. It's the cytotoxic T cells or CD8 T cells and helper T cells. Helper T cells have lots of subgroups, and they regulate the immune response in general. They can направить either directed to pro-inflammatory or anti-inflammatory direction or some moderate direction. And cytotoxic cells, they just kill the infected cells or malignant cells. And they act via recognition of antigens, processed antigens in the complex of major histocompatibility complex. Okay? But there are some interesting subgroups that are kind of not unique, but well, they are resident for the intestine, and they are mainly live and reside in the intestine. That's the gamma delta T cells, which are the common T cells. They have the receptor of two chains, that's alpha and beta. I said that they suffer some somatic recombination, and they can change the block of genes, which comprises their receptors. And this is their TCR, T-cell receptors are made of gamma-end chains. These chains are of restricted specificity. They can recognize antigen through MHC complex or without it. But mainly, they recognize the MHC independent antigens, phosphoantigens, for example, some components of the terpenoid synthesis pathway of bacteria. And they have lots of functions. They are some kind of innate regulatory cells. They can orchestrate all the immune response because we have to have the assurance. If something goes wrong in the adaptive lineage, we have to be sure that innate lineage can do at least something. Innate natural killer T cells. They have both the T cell receptor and the NK receptors. They can recognize some conservative antigens that recognize the natural killer cells, and they can recognize the antigens via the MHC component, but that's the non-classical MHC CD1 because their T cell receptors are restricted in specificity too. So they recognize hydrophobic peptides and some oligosugars of the bacterial cell walls. And they have the immune regulation function and the cytotoxic function. Immune lymphoid cell line, they do not express the TCR, they express just NK receptor and some conservative receptor for antigens like toll-like receptors and non-like receptors. And they are the immune regulations from innate range 2. Well, if we can say that the NK cells is NK, just NK, not the INK T cells, is the insurance that cytotoxic response will work, even if the cytotoxic lymphocyte don't work. The ILCs is the insurance that the immune regulation will work, even if the T helper cells don't work for some reason. And the B1 cells. The usual B cells are B2 subpopulation. They have the somatic recombination, somatic hypermutation. They make some specific antibodies, and they are origin from the bone level. But B1 cells origin from the embryonal linear, and they reside in the intestine in the early development, early embryonic development, and they don't need the antigen to produce the antibodies. They just produce the low specificity antibodies all the time, just to bind all the antigens that we can have. That's the first line of defense. Well, how does it work in general? What receptors are working here, and what parts of microorganism forwards the specific, or not specific for now, immune response to the microorganisms in the intestine. Well, toll-like receptors, there are lots of them. You can see that's the number 2, number 4, number 9. Between 4 and 9, there are at least 3. And between 4 and between 2 and 4, and between 4 and 9, there is 5, 6, 7, 8. There are lots of them, but these are the main for the recognition of some microbes that may be in the intestine. Well, TLR2 recognizes the lipoprotein acid, which is the component of the gram-positive cell wall. TLR4 recognizes the lipopolysaccharide, which is the component of the gram-negative cell wall, and some feature proteins, which are different in bacteria than in eukaryotic cells. And TLR9 recognizes the bacterial DNA. Okay, TLR2 drives the anti-inflammatory responses. It down-regulates the kappa-B MAP kinase pathways. TLR4 is opposite. It's the pro-inflammatory pathway. But if you think that's too easy, TLR9 can act both in pro-inflammatory and anti-inflammatory pathways, depending on some additional consequences. And okay, there are brif. There is a brif. Okay, that's not like protein, it's not only the free. And it recognizes the bacterial DNA. CpG, I haven't mentioned. Bacterial DNA has far less level of methylation of CpG islands in their genes promoters. So that's some kind of conservative issue of all the microorganisms. And such conservative receptors like toll-like receptors can recognize this level of methylation. So they do not react to the eukaryotic DNA, but they react to the bacterial DNA and mitochondrial DNA. That's just interesting fact. And NLRP3 reacts to the bacterial DNA, such as TLR9, to the late lipoteichoic acid as the TLR2, which is anti-inflammatory, and provides the pro-inflammatory immune response. So the meaning of this slide is just to understand that the variation is very complicated. Yes?
Can we take a question right now?
Yeah.
What type of textual proteins take part in this interaction with CR4? Is it microorganisms?
Yes, so that's microorganisms textual proteins. They have, I don't know the microorganisms textual proteins classification for sure, but they have some homology with the eukaryotic textual proteins. But I believe something like that, yeah, but maybe smaller, because that's from the microorganisms.
Wait, am I understanding right that another key is not internal protein?
It's not the membrane protein, yes, it's inside of the cell.
So
how is that recognized? Okay, I will explain. Because cell phagocytizes the antigen, yeah, and it's in the phagosome. Then it's mixed up with the lysosome, and the non-one is in the membrane of the lysosome. So inside of this big bubble, not another free protein can recognize the antigens inside the cell.
yes, I understand. Why do we have to recognize the antigen if it's already in lysosomes? Isn't it
To drive some kind of immune response. To drive the response in the regulational pathway, to regulate the expression of genes of some interleukins that are pro-inflammatory and anti-inflammatory, to drive the expression of more receptors, TLR, of some kind. And that's far more complicated if we're talking about more fine regulation with the bacterial secretory molecules. Because on the previous slide, we were talking about the main components of the bacterial cell wall, of their proteins and etc. But bacteria secrete lots of molecules as co-products of their living and not co-products. And they all can regulate the immune response. They all can interact with the immune cells and the certain receptors. For example, the vitamin A, which then becomes the vitamin acid, and it can regulate the B cell antibody secretion. It communicates with it blocks the histone deacetylase proteins, and so it becomes possible to regulate some expression of certain genes on the level of transcription. And Well, that's not on this scheme, but retinoic acid drives the T lymphocytes to the T regulatory pathway. There are lots of SCFAs that are working as the histone deacetylase inhibitors as well, and they bind to the certain receptors for the SCFA, such as G-coupled proteins. And okay, there are lots and lots and lots of secreted molecules that regulate the immune response. That's the meaning of this slide. I don't want you to know any of it. And what is BRIF-2? In the previous slide, I just wanted to say that the interaction between microbiota and the immune system of intestine is very complicated, and it may affect the immune responses very much. And that starts from our birth, because when we're going out naturally without some cesarean resection, etc., we have one microbiota composition. But if not so natural, with a surgical help, we have another microbiota composition, and that may lead us to the different development of the intestinal immune system and the development of certain diseases. You can see that, okay, you may not see it because maybe you can. Okay, I will do it. Yeah. If the intestine is normally organized, we can see the normal development of all the intestinal immune system. We see the normal pairs patches with lots of cells and lots of interactions. We have lots of pathways of differentiation of T cells and normal transfer of antigens to the mesenteric lymph nodes. But okay, if there is some dysbiosis, which is happening after the birth from cesarean section, or because of using of antibiotics and certain treatment of other diseases, there is far less plasmablasm.
Diverse?
Yeah, far less diverse composition of microbiota, and that's lead us to the unnormal development of the intestinal immune system. For example, there is a restricted development of subpopulations of T lymphocytes. The battery is off. Okay, I will use mouse. Sasha, I think it's smarter than you. You cannot fix it.
No,
I can do what? I didn't understand.
No, I think that is a noun. There is
Non-removable.
Yes, non-removable, and there are no two screens.
Yeah, there's just one screen on two screens. One screen on two screens.
Okay.
Well, that's not what I expected, but we have what we have. And that dysbiosis leads us to the certain and autoimmune diseases. There are lots of them. I will just concentrate on the inflammatory bowel disease just because I don't have so much time to speak out about everything. There is a brain-gut axis that is very, fashionable way to make some research on the theme of intestinal immunity, how it regulates our mind and our psychiatric conditions. But, well, inflammatory bowel disease is chronic inflammation in the intestine. There are two types of it, depending on the location of inflammation, but it's not so important. You can see that there is some environmental factors such as antibiotics and stress and etc. And it dysregulates the immune response, and it leads us to the excessive inflammation and intestinal bowel disease. Well, genetics is very important too, but I just talked in general. There are more than 20, I believe, mutations that are associated with the elevated risk of inflammatory bowel disease, including one of them is the mutation in the CART domain of node-like receptor. And these factors, environmental factors, can lead us to the changing of the microbiota substitution, usually like not the general phylum Firmicutes and Bacteroides are in the certain balance, but this balance could be destroyed because of some additional factors, and that drives the normal regulation by the T-rex cells development. But if there is damage to this equation between Firmicutes and Bacteroidetes, there will drive the immune response towards the Th17 pathway, which is some moderate immune response pathway, which is not very pro-inflammatory like Th1 and not as not pro-inflammatory like Th2, something at the border, but it drives the immunity at the state of chronic inflammation, and that's what happens in lots of autoimmune disease. Otherwise, these groups of microorganisms usually downregulated in the state of some dysbiosis. These campylobacters, bifidobacteria, lactobacilli, even Escherichia coli, they are downregulated in the case of dysbiosis. And so that leads us to the blocking of the T-regulatory cell pathway and the differentiation of pro-inflammatory phenotype. So, in conclusion, immune regulation of intestine is very complex, and it depends on the microbiome substitution. So we're just starting to elucidate some laws in this field, and there is a lot to know for us, but we can know for sure that intestinal inflammation is something that is something unique, something that we can't just meet in all of the tissue, and it depends very much. It depends very much on the microbiota substitution, and yeah, lots of projects I know in SCAM2 is toward to know these differences when the microbiota is healthy, when the microbiota is not very healthy, and how does it affect the immune regulation. That was just a brief overview of how does it work in general. So thank you for your attention. If there is any additional question, I can assume that that was not very easy to precipitate. No, precipitate. Okay. Just ask the questions.
There are some text in the chat. Maybe you should account this for receive message.
Sounds new. Okay. That's too late for this. I have one simple question about leaky gut, and did you come across an article about microplastics causing toxicity in intestine? Well, I don't have a good news for you because I think I don't have much of the information about this to answer this question. No, I don't know about the microplastic causing the toxicity in the intestine, unfortunately. Maybe some other questions?
Yes, I have a simple question. Why are there so many ways for name our words? I noted intestines, gut, colon, bowel, dreads, when you have so many names for it.
So many names for the intestine. Okay. That's
between colon and bowel and dread.
they are located in different sites. The colon is the one type. There are lots of colons actually. There's duodenum, sigmoidal colon, and etc. That's just an anatomy. That's different. I don't know how to say locations of the intestine when you're talking about the one big intestine. About the leaky gut, okay. To be honest, I hear this term for the first time in my life. Leaky gut. Huh? Leaky. Syndrome раздраженного, it's inflammatory bowel disease. That's what I've been talking about. Leaky gut. That's some kind of diarrhea. Oh, I didn't heard about. Like which pathway is activated during intestinal toxicity? Well, all of the excessive inflammation leads to the intestinal toxicity. Any of them. Just Th1, it's some kind of direct rough inflammation. Th17 is the inflammation that is very long in time, but very damaging at the same time. And the Th2 pathway leads us to the antibody-dependent toxicity. They may have other symptoms, different symptoms, but any kind of excessive inflammation leads to the intestinal toxicity. Well, lots of drugs are developing now for inflammatory diseases of bowel. They even use the antibodies, engineered antibodies towards the pro-inflammatory cytokines, antibodies against the interleukin-1, interleukin-12, interleukin-6, interleukin-18, and lots of them. That's the most targeted therapy, but the most expensive. But you can use just some immune suppressors, such as corticosteroids, but that have some additional effects. It may cause cancer in the future. Did I answer the question? It may be some excessive engineered interleukins, such as interleukin-10, which is anti-inflammatory. But don't you think administering antibodies can cause multi-drug resistance in bacterial population? Why should it? That's the regulation of the immunity itself. We administer the antibody of certain specificity. This antibody is specific not to the bacterial antigens. It's the antibody towards the which works towards our interleukins, because the interleukins, that's what causes the excessive inflammation. So if we block the interleukin-6 or interleukin-18, which drives the inflammation, we can stop this inflammation. So not, I don't think so. That's not the antibody specific to the bacteria when we're talking about the inflammatory bowel disease. That's the antibodies specific to our own cytokines. Any more questions?
Okay. What do you think about scar therapy and have you any idea if you can improve it for both media and application?
I don't think. Okay. I don't think CAR T therapy can help at all because okay, CAR T therapy is made to destroy something, but we have to prevent the destroy of the intestine.
Is there any generic receptors for?
But the cells, the function of the cells are the same. Well, we can make some chimeric T regulatory cells. I heard about this, but they are very dependent on the context of certain site which they reside and which we applicate them to, because the pro-inflammatory environment can drive them to the pro-inflammatory state. They may dedifferentiate and redifferentiate to the Th1 state, and they won't help. They will just make more damage.
I have one question. It's connected with my research, and maybe some common question, but it's really interesting for me. We know that dysbiosis and microbiota itself can contribute in autoimmune diseases, and it is connected with Th17 immune response. Is that right?
As well as other THs. Th1 and Th2 can drive the
Autoimmune.
yes too. But Th17 is just far more common for this type of disease.
So, as an you mean that bacteria or some other microorganisms can induce the Th1, Th2, and Th17 inflammation immune response? But if we focus the first question, why, how it happens when immune system thinks that now it's something about autoimmune Th17 immune response instead of Th1 and Th2, because okay, microbiota, we can say that it's like some organ of our system, but it's still some other microorganisms. And how do you understand me?
I think so. I'm
sure that I understand me now. So I hope you.
The differentiation is the rough word for this process, but okay, I will use this. Differentiation of naive T cells towards the naive Th cells. Okay, I will say more correctly, naive CD4 positive T cells towards the Th1 pathway, Th2 pathway, or Th17 pathway, or even Th9 pathway, or even T regulatory pathway is very context dependent. So the priming, the interaction of T cell with antigen presenting cell is the same for all these processes, and the phenotype is changing just a bit, just in the case of expression of certain receptors, and it's dependent on additional signals which T cell is receiving during the antigen presentation. These signals are the co-stimulatory membrane-bound molecules such as CD28 and the cytokine microenvironment in certain lymph tissue. And so we can say if the cell receives more pro-inflammatory signals than anti-inflammatory signals, it may become the Th1 cells. If the more anti-inflammatory signals, it may become the T regulatory cell or Th17 cell.
And it is summed up by the
Okay, how do the microorganisms
microorganisms
and the microorganisms and their secreting molecules and the components of their whole cell, they affect the inflammation that is regulated by the innate immune system. So if there is more components of, there is more metabolites that are drive pro-inflammatory responses, there will be more pro-inflammatory cytokines surrounding the Th cell, which is interacting with the antigen presenting cell. If there is more metabolites, there are more metabolites around. So more metabolites which drives the anti-inflammatory response, innate response, then it would be more anti-inflammatory cytokines surrounding the Th cell, which is reacting with the antigen presenting cell. And these happenings, they all affect the fate of the naive T cell.
One more question. What about some metabolites? For example, you said about vitamin A, which is
Retinol. Yeah,
acid.
it can be
So
metabolites to retinoic acid.
it's like it's about vitamin A, which produced by bacteria. But what if we get some these compounds like vitamin A from eat or taking from environment?
To use it as a drugs?
So is it has it the same effects, or I think it's the answer is yes, but I'm not sure. So what's the difference?
I'm not sure I understood the question properly. Can you please repeat it in 2 words? In several words, okay.
Differences between microbial metabolites and the same compounds, but
synthesized, no
what-
difference. There is no difference. That's the question of concentration, just concentration. Yes, of course.
Can you please explain where immune cells, which you listed, are located inside the organism, near the intestine or far from the intestine, in the blood vessels, or where are they reside?
That depends. What these kinds of cells,
All kinds of cells.
These certain kinds of cells, types of
All-
cells,
all-
they
all-
all reside.
cells
They always reside in the intestine.
where
But-
in the intestine?
Where in the intestine? In the intestinal epithelium.
Here.
Yeah. Lots of they are commonly they and some antigen presenting cells. They can be at the lesions of the M cells. It's not very good to do. Okay, it's working. You see these lesions in M cells. That's for the antigen presenting cells and some other cells that can reside outside but inside the M cell. M cell can surround them. So they usually can reside in the epithelium itself, but usually they reside close to the Peyer's patches just because they are the centers of all immune response.
So they somehow go through the basal membrane, or
Yes, yes. They have the homing receptors and inflammatory. Okay, when we're talking about, there are residential immune cells which reside in the tissue from the embryonic state of development, and there is not a question of basal membrane. They just move because of their homing receptors. They still and they remain here for all of our lives. And there are some inducible cells which are commonly in the bloodstream, but when there is some inflammation of the tissue, they receive the signal of some chemokines, of cytokines, they start expressing certain homing receptors to the certain tissue, and they just go through the basal membrane from the bloodstream, and here we go, they are in the site of the inflammation. That's when we're talking about these cells. And these cells, they are residential. They are always in the intestine from the embryonal state of development.
And another question, would the immune cells somehow go inside the intestine? You understand?
In the lumen.
In the lumen, yeah.
Well, I think it's hard to imagine the functional mechanism of something like that. But maybe in the case of damage, they can be outside the intestinal epithelia. But okay, every immune cell is dependent on the surrounding, and it just cannot provide some response by herself. And in these words, their functional role in the lumen is elusive. I don't think they can do something like that. I don't think they can do something in the lumen of intestine. There is no question. There are no questions in chat. You have one, or you have additional questions? No?
I just wanted to say that in the last of the lecture, it's this box of the Zoom.
Just now. Okay. Nothing happened. Any additional questions, or we can just go and thank each other for our time. Okay, thank you very much. Thank all of you. You can, if you have some questions or you want to cooperate on some project, you can just contact me. Any of you. So some of you know me, some of you don't. So okay.